Hello students, welcome to the EPG Pathshala. I am Dr. Snelata Panwar from the School of Life Sciences, Jawaharlal Nehru University, New Delhi. Today I am going to talk about the module Classification of Lipids, Simple and Compound Lipids, Phospholipids, Cholesterol, etc. from the paper Biomolecules and their interactions. So the learning objective of this particular module is basically to introduce you all to various subtypes of lipids such as simple lipids, compound lipids, and derived lipids, and to understand as to how these various classes of lipids function in the cell. So lipids can be classified as simple lipids if they are esters of fatty acids with glycerol and monohydric alcohols. So the simplest simple lipid is basically a triacylglycerol. Fats and oils are also classified as simple lipids because they contain triacylglycerols. Waxes are example of simple lipids. The next subtype of lipid is compound lipids, where the esters of fatty acids and alcohol contain other groups also. So basically, the basic structural unit in all these subtypes of lipids is a fatty acid, and based on how these fatty acids are conjugated to other groups and based on what kind of groups they are complexed with, these are classified as simple and compound. So compound lipids would be something like phospholipids, glycolipids, lipoproteins, etc. The third subtype of lipid is derived lipids, which are basically compounds of hydrocarbon rings and a long hydrocarbon side chain. So one example of derived lipid is cholesterol. Another example is steroid hormones. So more examples of various lipids would be like triacylglycerol, ceramide, waxes, etc. They're all simple lipids. Lipoproteins, glycolipids, phospholipids, sphingolipids, sulfolipids, etc. are all compound or conjugated lipids. Cerebrosides, gangliosides are a subtype of glycolipids. Glycolipid is a kind of conjugated lipid. Derived lipids would have cholesterol, steroid hormones, fatty acids, eicosanoids, etc. in this particular subtype. So let us have a look at one of the simple lipids, which is triacylglycerol. What are the functions of triacylglycerols? Triacylglycerols are also called triglycerides, fats, or neutral fats, and are considered simplest lipid molecules. We've already seen that a triacylglycerol molecule has a single glycerol molecule in ester linkage with 3 fatty acids. Tripalmitin, tristearin, etc. are considered simple tags as they contain the same kind of fatty acid, while mixed tags have 3 different fatty acids attached to the glycerol backbone. This shows how fatty acid is linked to glycerol through an ester linkage in order to give rise to a triglyceride molecule. And like I've already mentioned, that if the 3 fatty acids are identical, it's a simple tag. If the 3 fatty acids attached to the glycerol backbone are non-identical, it's a mixed tag. Let's have a look at the characteristics of tags. So tags are non-polar molecules, as the ester linkage between the carboxylate group of fatty acid and the hydroxyl group of glycerol leaves no free polar group. So these molecules are non-polar. Lipids in general have low specific gravity as compared to water. Therefore, oil will always float on top of the aqueous phase in a mixture of oil and water. Tags, microscopically, if you look into the cell, will appear as oily droplets in the aqueous cytosol. Adipocytes or fat cells are specialized cells which are basically the sites for synthesis and storage for tags. Cells other than adipocytes may contain one or few small oil droplets in the aqueous cytosol versus adipocyte, which is entirely filled with flat globules. So tags not only serve as energy source in humans, but tags also provide energy during seed germination. So both adipocytes and seeds are rich in enzymes called lipases. These lipases catalyze the breakdown of tags, which then mobilizes tags for energy production. Triacyl glycerols are also called as energy depots because they function as energy reservoirs. It is advantageous for the cell to have triacyl glycerols as energy reservoirs because of two reasons. First, the triacyl glycerols are less oxidized compared to proteins or sugars. Thus, they yield more energy per unit mass as compared to a molecule of glycogen. Secondly, being hydrophobic in nature, triacyl glycerols are stored in an anhydrous form as opposed to glycogen, which is normally stored in its hydrated form. Therefore, a breakdown of triacyl glycerol yields about 6 times more energy than a hydrated glycogen molecule upon complete oxidation. So besides serving as energy depots, tags also serve as insulators in animals like seals, penguins, and are very important for hibernating animals. In hibernating animals, the store tag serve two functions: as an energy source and as an insulator. So the low density of tags enables sperm whales to match the buoyancy of their bodies to match that of the surroundings during deep dives for food. So these tags are basically present in the head of these sperm whales. These sperm whales, they need to dive deep into the sea in order to feed on their food source. So how do tags help these sperm whales during these deep dives? So sperm whales basically have a head which is made up of spermaceti organ, which contains about 3,600 kgs of spermaceti oil. This spermaceti oil is actually a mixture of triacyl glycerols and waxes with lots of unsaturated fatty acids. And this oil basically starts to crystallize at 31 degrees Celsius and is a solid at very, very low temperatures, which is what the sperm whale encounters when it dives deep into the sea for its food. So this crystallized spermaceti oil enables the whale to remain floating during the deep dives, waiting for squids, which are basically their food source. So let's have a look at the other type of simple lipid, which is called wax. Waxes are simplest fatty acid esters that occur in nature and structurally are esters of long chain saturated and unsaturated fatty acids with long chain alcohols as you see in the structure that has been shown here. This structure is of a compound called triaconithinyl hexadecanoate, which is obtained from beeswax. Waxes are also a storage form of energy in planktons. The melting points of waxes are in the range of 60 to 100 degrees Celsius, which is higher than the melting points of tags. Due to the water repellent properties of waxes, certain skin glands generate waxes to protect hair and skin, while preen glands that are present in birds secrete waxes to protect the feathers of birds from water. The leaves of plants like poison ivy, rhododendron, etc. are also waxy, which keeps loss of water in check and protects these plants from parasites. Let's move on to compound lipids. Glycerophospholipids or phospholipids are a type of compound lipid. These phospholipids are major components of the plasma membrane of any living cell. They consist of a glycerol 3-phosphate backbone with the C1 and C2 of glycerol in ester linkage with fatty acid. The phosphate group is linked to a polar head group, which is referred to X. And because these phospholipids have fatty acids as well as a phosphate molecule, they are considered as amphiphilic molecules with nonpolar fatty acids and polar X group. X can be H or it could be any other molecule, as we will subsequently see as we move on. If X is H, it is called phosphatidic acid, which is the simplest phospholipid that is present in biological membranes. So glycerophospholipids, I said, are amphiphilic. They're amphiphilic because they have non-polar fatty acids as well as they have polar head groups. And I said that the polar head group is depicted as X, and this X can either be H. If it is H, it is a phosphatidic acid. If the X group is a choline moiety, it is called phosphatidyl-choline. If it's a ethanolamine moiety, it's called phosphatidyl-ethanolamine. So you look at this structure that has been shown on the left side of the slide. There is a glycerol backbone. To the C1 and C2 of the glycerol backbone, there are fatty acids that are attached. There is a phosphate group that is attached to carbon 3 of the glycerol backbone. And this phosphate group is then attached to the polar head group. In this structure that has been depicted here, the polar head group is choline. That is why this molecule is called as phosphatidylcholine. This X or the polar head group can be ethanolamine. It can also be an inositol molecule, or it could be a serine molecule. The C1 position of the glycerol backbone is usually occupied by a saturated C16 or C18 fatty acid, while the C2 position is normally occupied by an unsaturated C16 to C20 fatty acid. Because of the presence of the phosphate group, there is a negative charge on a phospholipid molecule, while the charge of the head group can vary from one phospholipid to the other. As an example, in phosphatidylinositol 4,5-bisphosphate, it is negatively charged, while it has a positive charge in case of phosphatidyl-ethanolamine or phosphatidylcholine. If the head group is serine, then phosphatidylserine is neutral with no charge. So we have seen that fatty acids are normally linked to the glycerol backbone in phospholipids through an ester linkage. Besides that, there are also phospholipids which have ether-linked fatty acids. So ether lipids have one of the acyl chain in ether linkage to glycerol instead of the ester linkage. In the alkyl ether lipids, the ether-linked acyl chain may be saturated or may have a double bond between carbon 1 and carbon 2 as in plasmalogens. The hard tissue of vertebrates and the membranes of halophilic bacteria are enriched in plasmalogens, which is a very good example of an ether-linked fatty acid. Phospholipases are unable to cleave the ether-linked fatty acids like plasmalogens, which may be important for their roles in the hard tissues. Platelet activating factor is another example of a phospholipid which has ether-linked fatty acid, which is a signaling molecule that is released from leukocytes. It stimulates platelet aggregation and serotonin release from platelets. Phospholipids can be broken down by enzymes called phospholipases, and the products that are released after hydrolysis by phospholipases can actually help in determining the chemical structure of phospholipids. As you can see here in this uh, diagram, the sites of action of different phospholipases. So there are phospholipase A1, there is a phospholipase A2, phospholipase C, and phospholipase D. And all of these phospholipases act at different sites within the phospholipid molecule. So as an example, if a phospholipid molecule has been digested with phospholipase A2, the remaining molecule that is left is called lysophospholipid. Lysophospholipids function as detergents. Because they function as detergents, they have the ability to disrupt cell membranes. Bee and snake venoms are extremely enriched in phospholipase A2, and that in fact tells you why bee and snake venom are harmful to humans. We have seen that phospholipases are used to cleave the phospholipids in the cell. These phospholipases are important because they are enzymes that catalyze the breakdown of phospholipids. So basically, they are proteins, but in order to get an access to their substrate, which is the phospholipids, they have to enter into the highly hydrophobic core of the plasma membrane. So solving the crystal structure of these phospholipases has fascinated biochemists for a long time. And amongst all the phospholipases, phospholipase A2 is one of those lipid-specific enzymes that has been well studied. This phospholipase A2 is a small protein with 125 amino acid residues, about 14 kilodaltons molecular weight. The X-ray structure of this particular phospholipase from cobra venom has been obtained, which shows that the polar head group of the phospholipid fits into the active site of phospholipase A2, while the hydrophobic tails extend out from the active site to interact with many aromatic side chains. The hydrolysis products that are generated after the phospholipases have acted upon phospholipids generally do not undergo further degradation. For example, lysophosphatidic acid, which is generated in vivo in the membranes of platelets, does not undergo further degradation, but instead serves as an inducer of cell growth during wound repair process in the damaged cells. 1,2-diacylglycerol is another end product that is normally derived from the hydrolysis of membrane lipids by the action of phospholipase C, as we have seen before. And DAG usually serves as an intracellular signaling molecule that activates a protein kinase. Let's move on to another type of compound lipid, which are called as galactolipids. So galactolipids are also important constituents of cell membranes, but they are predominantly present in the plant cell membranes. So what do galactolipids look like? Galactolipids structurally consist of one or two galactose residues that are connected by a glycosidic linkage to C3 of diacylglycerol molecule. As shown in this structure, you can see that carbon 1 and 2 are occupied by fatty acid, while carbon 3 is attached to a single galactose residue. The thylakoid membranes of the chloroplasts are enriched in these galactolipids. Galactolipids are considered to be the most abundant membrane lipids in the biosphere. Plants also contain sulfolipids, which are another kind of compound lipids, where a sulfonated glucose residue is joined to a diacylglycerol in glycosidic linkage. Sphingolipids are also a type of, like phospholipids, are important constituents of the plasma membrane and help in providing structural integrity to the plasma membrane. Sphingolipids differ from phospholipids in the sense that they do not contain glycerol and are derivatives of the C18 amino alcohol, which is called sphingosine. So, sphingolipid contains the following: one molecule of sphingosine or one of its derivative, one molecule of a long chain fatty acid, and a polar head group joined by a glycosidic linkage in some cases and a phosphodiester in others. In the sphingosine molecule, which is the backbone of sphingolipid, the C1, C2, and C3 carbons are analogous to the 3 carbons of glycerol in phospholipid, as you can see in the structure here. The C1, C2, C3 are analogous to the 3 carbons of glycerol in phospholipids. If a fatty acid is in amide linkage with the amino group of the C2 carbon, it forms a ceramide. The X, which is attached to the C1 carbon of the sphingosine backbone, if that X is an H, then this molecule is called ceramide. Ceramide is the parent compound of sphingolipids. Sphingolipids can further be divided into different subclasses like sphingomyelins, cerebrosides, and ganglioside. Sphingomyelin comprises of about 10 to 20% of plasma membrane lipids, and structurally, sphingomyelin basically has the X group replaced by a phosphocholine or phosphatidyl ethanol moiety. So if you look at the structure here, you have the sphingosine backbone, you have a fatty acid in amide linkage with the amino group at the carbon 2 position of sphingosine, and the X here in the sphingosine backbone is replaced by either phosphocholine or phosphatidyl ethanolamine. So chemically, sphingomyelins differ from phosphatidylcholine or phosphatidylethanolamine, but they still share the same charge distribution and conformation. So sphingomyelin is an important constituent of the myelin sheath that surrounds nerve cell axons. Another subtype of sphingolipid is called cerebroside or glycosphingolipids. Cerebrosides are basically ceramides that contain a single sugar residue as the head group. As you can see in the structure, the sugar residue here is glucose. Examples of cerebrosides are glucosyl cerebroside or galactosyl cerebroside that are the most prevalent glycosphingolipids that are present. These cerebrosides are non-ionic because they do not contain a phosphate group and hence are also referred to as neutral glycolipids. So basically, glycosphingolipids or cerebrosides can have either glucose or galactose, but at a given time, they will only have one sugar residue attached to the sphingosine backbone. Gangliosides, as opposed to glycosphingolipids or cerebrosides, are more complex cerebrosides which comprise of ceramide with attached oligosaccharides that include at least one sialic acid residue. So you can see the structural formula of a ganglioside with its sphingosine residue in Fischer projection here, and you see that there is a single sialic acid residue that this particular structure has, or this particular ganglioside has. The head groups of gangliosides extend beyond the cell membrane. Because they extend beyond the cell membrane, they function as receptors for certain hormones. Gangliosides serve as determinants of cell-to-cell recognition. Thus, they play a role in growth and differentiation of tissues and in carcinogenesis. So the importance of gangliosides is highlighted by the fact that if there is a disorder in the breakdown of ganglioside, it results in a disease called Tay-Sachs disease. Now, the presence of at least one sialic acid residue is a must for ganglioside, but they can have more than one sugar residues. So, if the ganglioside has a single sialic acid residue, it is called as GM1. M refers to mono and G refers to ganglioside. So, it is referred to as ganglioside monoseries. If the ganglioside has two sialic acid residues, it belongs to a GD series. D stands for di. And if it has more than two residues, like for example, if it has three sialic acid residues, the ganglioside is classified as a GT ganglioside, which means ganglioside tri-series, and so on and so forth. Here we see a flow chart of glycosphingolipid classification. So far we've seen that glycosphingolipids can be neutral or acidic. Neutral glycosphingolipids are further classified as cerebrosides or globosides. Cerebrosides are the ones that have a single sugar residue like glucose or galactose. If the cerebroside has a glucose residue, it's a glucose cerebroside. If it has a galactose residue, it's a galactocerebroside. The other neutral glycosphingolipids are called globosides. Lactosyl ceramide is one of the examples of a globoside. Gangliosides and sulfatides are classified as acidic glycosphingolipids. As we've seen, gangliosides contain an acetyl neurominic acid, which is also called as sialic acid, which imparts a negative charge on these gangliosides and therefore making them acidic. Sulfatides are cerebrosides with a sulfate group, which also imparts a negative charge to these glycosphingolipids, and hence they're classified as acidic glycosphingolipids. Let's now move on to the third subtype of lipids, which are called as derived lipids. The steroids are examples of derived lipids. They are derivatives of a compound that is constituted of 4 fused nonpolar rings. 3 of these rings have 6 carbons, as you can see in the structure that is shown below in the slide. 3 of the rings have 6 carbons, while one of the ring has 5 carbon. Cholesterol, like phospholipids and sphingolipids, are required by the eukaryotic cell membrane because they also provide structural integrity to the cell membrane. The fused rings, also referred to as the steroid nucleus, are planar and rigid, and so do not allow rotation around the carbon-carbon single bond. Cholesterol also, like phospholipid and sphingolipid, is an amphipathic sterol molecule because it has a polar head group in the form of the hydroxyl group that is attached to one of the rings in the 4 fused rings. So this polar hydroxyl group provides it polarity, while the fused rings along with the alkyl side chain provide the non-polarity to the cholesterol molecule, and therefore cholesterol is considered to be an amphipathic molecule that provides structural integrity to the cell membrane. Bacteria cannot synthesize sterols but can incorporate exogenous sterols into their membranes. Sterols are important because they serve as precursors for steroid hormones and bile acids. Bile acids, we all know, are actually polar derivatives of cholesterol that function as detergents to emulsify fats in the intestine for a better access to lipases. So, steroid hormones, based on their physiological response they are involved in, can be classified as glucocorticoids, mineralocorticoids, androgen and estrogen, and vitamin D. Cortisol, a C21 compound, is a glucocorticoid, while aldosterone is a mineralocorticoid. Testosterone and estradiols are androgens and estrogens, and vitamin D is also a steroid hormone. So depending on the physiological response, we see that the steroid hormones can further be classified into different subtypes. So let's just have a look at what are the different functions that these steroid hormones perform in the human body. Glucocorticoids are synthesized by the cortex of the adrenal gland. They affect carbohydrate, protein, and lipid metabolism. They control inflammatory reactions, and the impaired functioning of glucocorticoids results in Addison's disease. Mineralocorticoids like aldosterone are involved in the regulation of the excretion of salt and water by the kidneys, while the androgens and estrogens like testosterone and estradiol are male and female hormones. Testosterone is synthesized by the testes, while the estradiol is synthesized by the ovaries. Let's have a look at the functions of vitamin D, which is also a steroid hormone. Vitamin D regulates calcium metabolism. There are various forms of vitamin D like vitamin D2 and D3. Both vitamin D2 and D3 that are generated in the cell are inactive. The active forms of these vitamin D isoforms are produced through their hydroxylation, which is carried out in the liver. So active vitamin D increases the absorption of calcium from the intestine, which is obtained basically from the diet. This results in deposition of calcium in teeth and bones. The deficiency of vitamin D causes rickets in children, we all know that. And due to its property of being water soluble, an excess intake of vitamin D is also harmful because that can cause vitamin D intoxication. So we've now seen how important the class of derived lipids are because they serve important roles in the human body, like we've now seen as to how the different kind of steroid hormones are required for performing different functions in the cell. So what is the take-home lesson from this particular series of lectures? Lipids are classified as simple, compound, and derived, which are further divided into various subtypes, as we have seen. Triacylglycerol contains 3 fatty acids that is esterified to the glycerol backbone and are stored in specialized cells called adipocytes. Glycerophospholipids are conjugated or compound lipids, which are basically amphiphilic molecules because they have a polar head group and 2 fatty acid chains in an ester or an ether linkage to the glycerol backbone. Sphingolipids are also considered as compound lipids, which include sphingomyelin, gangliosides, and cerebrosides. Glycerophospholipids and sphingolipids both provide structural integrity to the biological membranes. Cholesterol, on the other hand, is an example of a derived lipid which has a 4-ring structure. Steroid hormones and vitamin D are all based on this 4-ring structure and serve important biological roles in the cell. So basically, simply put together, we have seen that lipids can be divided into various classes and various subtypes. All these subtypes serve important functions in the cell in the sense that they provide either structural integrity or they serve as intra or intercellular signaling molecules. Thank you.